Today, we'll be talking about gel electrophoresis. What is gel electrophoresis, you might ask? Well, it's a lab technique usually used in the biochemistry lab for separating out DNA or proteins based on their size. And let's talk about how it works. So first you need to have the gel. This can be made out of different kinds of substances such as agarose and polyacrylamide, both of which I'll discuss later. And the electrophoresis part of it means that you need to have an electrical field passing through the gel to get the bands to move. So to create an electrical field, we have to have a cathode and an anode. The cathode is on this side. Remember from general chemistry that at the cathode, reduction takes place, so you'll have a negative charge. But on the other side, where the anode is, you'll have a positive charge because this is where oxidation is taking place. And to power this, you'll need some kind of battery to connect them. And these are both usually also connected to a big box that contains the gel electrophoresis apparatus. In order for charge to flow across, you need something that will conduct electricity. So you have a buffer that's usually composed of different ions. So this is completely covering this entire gel. And remember that it's always there, but I'm going to erase it just because it's going to get a little messy for what I need to show you next. So next, you'll load a sample of DNA. In order to load your sample, you'll need to take a pipette and put it into one of these wells. So usually you'll want to make sure that you have a dye mixed in with your sample so that you can see it as it's running. So I'll show that as a pink line here, so I've filled that well, followed by yellow here, and green here. So that's just the color of the dye. It can really be any color you want. This will just help you track the movement of the bands later on. Once you have these bands, what will happen once you turn on the electrical field? Remember that DNA has a negatively charged backbone because of all the phosphate groups. So what you'll actually observe is that they'll travel towards the positive end. Okay, so maybe after a short period of time, what you'll see is that it'll look something like this. Maybe the pink will have split up into a few bands, indicating that there's a few different size fragments in there. With the yellow, you might only see one band, and with the green, you might actually have two bands, but they're so close together still that it's kind of hard to tell. So how can you really see what's going on? The solution is to let this run for a longer period of time. And eventually what you'll see is shown here at the bottom. You'll see that whatever size fragments were in your original samples have effectively split up by their size. And note that in pink, you'll always want to load something known as a DNA ladder. A DNA ladder is like a standard. This is something that you buy from a company, and they tell you exactly what sizes their fragments are so that you can match them up to your unknown samples. So this could be 400 base pairs, 200 base pairs, and 100 base pairs. And as you'll note, the smallest fragments travel the furthest. This is because the smallest things are really easy to push with the electrical field. But when you have such a big molecule, or rather a big DNA fragment, it can be hard to move. So you can see that the 400 base pair doesn't move too fast or too far. And what does this tell us about our unknown yellow and green samples? This shows us that the yellow sample has a band that's 200 base pairs long, and the green sample actually was composed of two different fragments, one that was 100 base pairs and another that was 200 base pairs.
How does DNA fingerprinting work? If you've ever seen CSI, or one of those reality talk shows where a man demands a paternity test for a baby he doesn't believe is his, you'll have seen the two most common applications of DNA fingerprinting. But what's actually involved in the process? First, let's look at how DNA fingerprinting was discovered. On September 10th, 1984, at 9:05 in the morning, Alec Jeffreys had his eureka moment. His team at the University of Leicester had been working for 7 years to see if it would be possible to tell people apart and whether they were related using their DNA. They'd been looking at regions of the human genome called VNTRs, which stands for Variable Number Tandem Repeats. These can be thought of as a repeating string of genetic letters or bases, a bit like the same word written down many times along a line. In this case, the words are between 10 and 80 bases in length, and the numbers of repeats of the word can be as high as 30. You inherit these repeated sequences, along with the rest of your DNA, from your parents. But because the VNTRs occur within parts of the genome called non-coding regions that aren't actually used to encode proteins, genetic changes called mutations and duplications, which are copies of the DNA sequence, take place within the VNTRs much more often than in other parts of the genome. This means that a person is likely to have a similar number of repeats to their close relatives, but people more distantly related to them and complete strangers will have a totally different number and therefore different length VNTRs. It's these differences that can be exploited to tell one person from another genetically. On that morning in 1984, Jeffries was looking at the results of an experiment they carried out to see if a probe they'd created to pick up one particular VNTR sequence could show up variations in the length of the sequence repeats in the DNA of his lab assistant and her parents. A clear pattern was visible, confirming that she'd inherited repeats of a different length from each parent, and the world of DNA fingerprinting was born. After this breakthrough, Jeffries' lab was the only one to offer DNA fingerprinting, and they were in high demand with cases from all over the world until the process was commercialized in 1987. Nowadays, rather than VNTRs, DNA fingerprinting makes use of sequences called short tandem repeats, or STRs. These are similar to Jeffries' VNTRs, but are smaller and stand up better to the degradation of DNA over time, which makes them much more useful for investigating crime scenes. So let's look at an example of how DNA fingerprinting is carried out today. The DNA from a sample such as a mouth swab, blood sample, or a swab from a crime scene is extracted and purified. In order to isolate the short tandem repeats, a procedure called PCR, or the polymerase chain reaction, is carried out on the DNA. PCR copies or amplifies specific target DNA sequences. In this case, the STRs are targeted, increasing their relative numbers in the sample. The different length STRs copied by the PCR reaction are then separated out using a process called electrophoresis. This involves adding the DNA to an agarose gel and passing an electric current through it. Because DNA is negatively charged, it'll move through the gel towards the positive electrode. The larger, longer STR repeats, which are made up of longer strings of DNA letters, are heavier, so they move more slowly through the gel than shorter, lighter repeats. And so by running the electric current for a certain period of time, the different-sized pieces of DNA will spread out through the gel. They can then be revealed using a dye to label them up, producing a line called a band on the gel. The pattern of these bands showing the presence of different size repeats is the genetic fingerprint of a person.